Welcome to our first lesson on cell biology. In this unit, we will learn three important topics. The first one is the theory of cell theory, and the second one is the types of cell, and the third one that we will discuss is parts of cell and their function. In this period, we are going to start from cell theory. To begin our discussion, we have to list out the objectives of the lesson. At the end of this section, students, you should have to able to tell the history of cell biology, and you have to describe cell theory and investigate the size, structure, and shape of cells. And lastly, you are able to state the basic functions of cell. To start the cell theory, we need to talk about the timeline for the development of cell theory. In this part, we are going to discuss the contribution of 6 biologists for the development of cell theory. The first biologist that we are going to see is Robert Hooke. Robert Hooke saw tiny structures under his compound microscope, and this compound microscope magnifies only 30 times. It magnifies only 30 times, and when Robert Hooke observes his sample under the compound microscope, he saw cork cells. He saw cork cells and tried to sketch the image under the microscope. And after sketching the image, he called these tiny structures cells. He called the structures cells. In addition to this, when Robert Hooke coined the name cell for these small, very small structures, he used the compound microscope, and at this time the cells are dead cells. The cells are dead cells because you know that cork cells are dead cells. And the next contribution for the development of cell biology goes to Anton van Leeuwenhoek. Anton van Leeuwenhoek was a lens grinder, and when he formed lenses with different magnification power, he observed different samples, and in his observation he saw drop of water, and when he observed under the simple microscope, he observed tiny moving and unicellular organisms. The drop of water contain these unicellular organisms moving here and there in the microscope, and he called these tiny moving organisms animalcules. He called these tiny organisms animalcules, and at that time Anton van Leeuwenhoek used a microscope that can magnify 300 times. But remember, the microscope that was used by Anton van Leeuwenhoek was simple microscope. I hope you know that simple microscope is type of microscope with only one lens. In this time, this simple microscope was used to observe the tiny organisms in drop of water. And in addition to this, Anton Leeuwenhoek took sample from his mouse, and he observed the sample, and he observed bacterial cells, and finally he called these bacterial cells animalcules. He called the bacterial cells animalcules. The Anton van Leeuwenhoek contribution for the development of cell theory is he was the one to observe living cells for the first time, and secondly, he tried to show different types of cells by his simple microscope. The third contribution was by René Dutrochet. This scientist or biologist concludes that all living organisms are composed of cells. All living organisms are composed of cells, and this is one of the contribution by René Dutrochet for the development of cell biology. In addition to these contributions, René Dutrochet provide additional discoveries for the scientific world. One of the discovery was stomata. He discovered stomata for the first time, and these structures are found on leaves of plant. And the other discovery was the process of osmosis. Osmosis, as you know, is a movement of water molecules into and out of the cell, And the other contribution of René Dutrochet was the importance of chlorophyll for photosynthesis. You know, from your lower grade, photosynthesis use chlorophyll for absorbing light energy, to change the light energy into chemical energy by green plants. The other contribution was René was the one to indicate that respiration occurs in all plant and animals, in all organisms like plant and animals. And he also concludes that all growth occurs because of increase in volume by addition of more little cells. These are the contribution by René Dutrochet. The next contribution goes to the Matthias Schilden, who was a botanist, and he concludes that all plants are made up of cells. He concludes this conclusion. At the same time, the other scientist, Theodor Schwann, who was a zoologist, you know that zoologists are scientists that study about animals, he also put the same conclusion, which says all animals are made up of cells. After having these two conclusions, they stated the first clearly stated cell theory with three important statements. The first statement of the cell theory is the cell is unit of structure, physiology, and organization in living organisms. The second statement says the cell retain a dual existence, which means the cell can exist as distinct entity or they can exist as building blocks in the formation of organisms. And the third statement of cell theory of Theodor Schwann and Matthias Schilden was cells formed by free cell formation. Cells formed by free cell formation. This is an idea that's supported by spontaneous generation. If you remember, on Unit 1, you have learned spontaneous generation, which states that living organisms arise from non-living organisms. This theory later on disproved by Louis Pasteur, and this idea is supported by spontaneous generation, which is raised by Theodor Schwann and Matthias Schilden. In 1855, the next biologist that contribute for development of cell theory was Rudolph Virchow. Rudolph Virchow was a physician who did research on cancer cells and states that all cells come from other pre-existing cells, which means this idea completely disproves the idea that says cells can be formed by free cell formation. Therefore, Rudolph Virchow states the most important idea in the development of cell theory. After that, the cell theory was modified by Rudolph Virchow by having these four statements. The first statement, Rudolph Virchow complete the first accepted version of cell theory, which says all organisms are made up of one or more cells, and all cells come from pre-existing cell, the second statement. And the third statement, the cell is unit of structure, physiology, and organization in living organisms. And the last statement is the cell retain a dual existence as distinct entity and as building block of organization. These statements are stated by Rudolph Virchow cell theory. Okay, up to now we have seen cell theories that were stated by different scientists at different time. But the scientific society state modern cell theory that has acceptance all over the world, and this modern cell theory include these 6 statements. The first statement, all known living things are made up of cells. And the second statement, the cell is basic structural and functional unit of all living things. And all cells come from pre-existing cell. And the other one, the fourth statement, cells contain hereditary information, which means DNA, which is passed from one cell to the other during cell division. And the fourth one, all cells have basically the same chemical composition. They have basically the same chemical composition, and all energy flow occurs within cells. These statements are statements that are considered as modern cell theory. Students, you have to remember that in this cell theory or in these statements, the modern cell theory rejects some ideas from previous theories and add some additional ideas to formulate the modern cell theory. So you are expected to pick these statements that are rejected in modern cell theory, but that were stated in previous cell theories. And the next one is, up to now we have seen the different cell theories, the modern cell theory, and so on. So now it's time to have a break in order to answer this question. We have 2 questions. The first question, the scientist who observed living cells for the first time was dash. And the second question, mention 2 statements from modern cell theory. You are expected to answer this question within 2 minutes. I have given you 2 minutes. Answer this question, and let's back to our discussion. The next point that we are going to see is the diversification of cells. Cells diversify in size, which means you can find variety of size of cells. Cells can diversify in shape, the same like that of size, they have different shape, and cells can diversify in their internal organization. When we say internal organization, it is to mean that the internal structures of cell vary. One cell vary from the other cell. Or in other words, if you take plant cell, they have completely different structural organization with that of animal cell. This is to indicate that cells vary in their size, shape, and in their internal organization. Let's start from their size. As we said, size of cell varies depending on the type of cell. But generally, when we look at size of cells, cell size can vary from very small bacterial cell to huge or large ostrich egg cell. This indicates there is variation in size between cells. That's why here it says the chicken's eggs are just one huge cell up to 5 centimeter or 0.05 meter in length. This is from large cell. And in the contrary, if you take small cells, the smallest bacterial cells are only just over 100 nanometer in length. This indicates there is variation in size between cells. So what is the secret behind having a different size of cells? This depends on the type of cells that we are going to measure. This is because if you take bacterial cells, they are generally small in size, so you can take measurements of bacterial cell by using small units. Or if you take large cells, you can use large units. But generally since these cells are small in size, we use 3 units that are small. And the first unit is millimeter, which is 1/1000th of a meter, or in your mathematics or physics or chemistry, you represent the millimeters as 10 raised to -3. And the second one is micrometer, even a smaller unit than that of millimeter is 10 raised to -6, and nanometer is 10 raised to -9. Imagine, these units are very small, so we are expected to measure cells by using these small units. You know the reason? Because cells are very small, so you are not expected to measure size of cells by kilometers, meters, and so on. So we have to measure size of cells by small units. Sometimes you may face different examples, or you may have different questions with different measuring units. At this time, you are expected to convert one of the unit into the other. Therefore, in order to convert these small units from smallest to the largest unit, we need to use one of these 2 mechanisms. The first one is to convert a larger unit to the next smaller unit. You have to bear in mind that to use this formula, the unit that you are going to change must be the next smaller unit. The next smaller unit, you simply multiply by 1000. This is very simple and elementary mathematics. And to convert smaller units to the next larger unit, what you have to do is you have to divide the number by 1000. Let's see example in both cases. Here we have example convert 3.5 millimeter into micrometer. Millimeter is the largest unit and micrometer is the smallest unit, the next smallest unit. So what we have to do, we have to multiply the given number by 1000 and we will find 3500 micrometer. And the second case, convert 87 nanometer into micrometer. It is the other way of conversion, a small unit into the next larger unit. In this case, we are expected to convert 87 nanometer into micrometer. You can simply divide by 1000 and you can find 0.087 micrometer. This is very simple. So we are going to measure cells under microscope, and then we have to put the size of the cells by one of these different conversions. Okay, students, we have seen a lot about unit measurement of cells and also how we can convert units from one form to the other. Here we have questions or exercise to convert one unit into the other, and also you are expected to do this exercise with 2 minutes. I will give you 2 minutes to calculate the answer for these questions. Okay, let's return back to our discussion. I hope you have calculated your exercise, and it is very simple. And the next point that we are going to see is what is the consequence of the different size of cell, or what will happen if all cells are large in size or all cells are equal in size, and so on. In order to answer this question, we have to compare different conditions. When a cell gets bigger, all its dimension will change. When we say all its dimension, it includes the volume, the surface area, and so on. Therefore, when the cell gets larger and larger, the volume of the cell will be changed, and also the surface area of the cell will be changed. That means if you change the size of cell, or if you double the size of cell, that does not mean you are doubling all the dimension of the cell. Some dimensions will increase, some will decrease. So in this part, we are going to see the relationship between surface area to volume ratio of a cell with its size, or we are going to relate if the surface area to volume ratio of the cell decreases or increases, related with increasing or decreasing the size of cell. But generally, what you have to remember is the surface area to volume ratio can be affected by increasing or decreasing the size of cell. Therefore, if you increase the size of the cell, surface area to volume ratio decreases. If you decrease the size of cell, surface area to volume ratio increases. You have to put this idea in your mind. And to have more clear idea about this concept, here I have provided you an example of a cube. A cube, as you know, is a structure with 4 equal sides, and you know it in your mathematics. And this structure has equal sides in all its direction, and here we are going to see the effect of increasing the size of a cube on the surface area to volume ratio. To understand this example, you are expected to see your textbook on page 180, figure 4.13. If you found the page, now let's see the concept of surface area to volume ratio with its relation with the size of the cell. On your textbook, the textbook provided you three cubes: cube A, cube B, and cube 3. If you look at cube A, it is very small and 1 unit length. And if you look at cube B, it is double with side of that of cube A, and the third cube is double with that of the second cube. By considering this difference in their size, now we are going to calculate the surface area to volume ratio. The surface area can be calculated simply by calculating length with width, but we have a cube, and cube has 6 faces, so we have to multiply 6 times the total area, and we can find the total surface area, and the volume is calculated by length times width times height. In this case, we have 1 unit length for the first cube, so the volume becomes 1 times 1 times 1, which gives 1 arbitrary unit cube. And the area that we have calculated in the above formula gives 6, and when we put the ratio of surface area to volume, the surface area is 6 and the volume is 1. If you look at the diagrams, they increase in size, and this increase in size finally leads to have decrease in surface area to volume ratio. From this, what you have to understand, or what you have to conclude, is as the size of the cell increases, the surface area to volume ratio also decrease. So what is the importance of having large surface area to volume ratio, or in other words, having a small size in cell? It is very important, because having a small size is important to provide the necessary material for the cell, and also for transport of materials within cells, and control of cell. If cells are very small, controlling mechanism become easy. And for metabolic requirement, when we say metabolic requirement, cells need requirements for their function. This can be supplied by the surface and can be processed by the volume. For that reason, cells must be small in size in order to have these 3 important advantages. Up to now, we have seen how we can measure cells and the importance of having a small size. This indicates as cells must be small in size so that they can build large organisms, and so that they can provide necessary nutrients. Okay, to wind up our discussion, we have discussed cell theory, development of cell theory, and in this part we have seen the contribution of 6 scientists or biologists. So you are expected to list their contribution. After having that, we have seen the statements of modern cell theory, which take some of the statements from previous theories and some updates. And lastly, we have tried to see cell measurement. In cell measurement, we have said that we need to use small units. This is because cells are very small, and to use small units, we have to know unit conversion. And when we convert 1 unit to the other, we have to follow 2 rules. One is multiplying by 1000 to convert into the next small unit, and the other one is dividing by 1000 to convert into the next smallest unit. And lastly, we have seen that the importance of surface area to volume ratio for cells. As we said, cells with largest surface area to volume ratio can provide all the necessary processes for cells as compared with cells with large size. And finally, we are going to see some questions based on our discussion. We have 2 questions. These questions can be your homework, and next time we will see the answer.